Record to the class. All right, so let's talk lipids. All right, this is going to be actually a 2-day lab, which means like Friday one lab period will do day 1, and then the next lab period will do day 2. So you kind of have a nice break in lab reports right now, which I think you kind of need because you're going to have a test and lecture, right? So I think it timed out pretty well. Yeah. All right. So if you want this in color, it's on Moodle. And the other thing that's really great on this is that there are no additional problems. There are no calculations outside of the calculation that I'm asking you to do within the lab report. But there are some things you have to do at the end, like in place of that, but it's not bad. Trust me, it's not bad. Okay. So there are 3 basic steps to extracting and quantifying phospholipids. The first one is you have to get the phospholipids out of your sample, and we're going to be using heart tissue. So I believe this semester we have, we're going to be using chicken hearts. You can use any heart, beef, whatever, anything, but I think this semester we're going to be using chicken hearts. So the first day is just extracting them, and then think about inside the cell, how many different lipids, just general lipids, do you have in the cell? A bunch of different kinds, right? One subset of those are phospholipids, right? So they have a phospho group on the head group, and there are variations of that. So what we're going to do is we're going to extract out primarily the phospholipids, then we're going to separate the phospholipids, and then on day 2 we're going to quantify the phospholipids. And when I say day 2, I mean like lab period 2, right? Okay, so let's talk about lipids in general. Lipids, I don't know why that happened. Does that show up on your paper too? Is your paper like that? No? Okay, so this is just this one. So lipids are insoluble in water. Why are they insoluble in water generally? They're very, part of them are very hydrophobic. So if you look down here, this is an example of a fatty acid, right? And lipids are composed of fatty acids. And so what portion of this is insoluble? The huge hydrocarbon tail. See if I could draw. The huge, this is the hydrocarbon tail of a fatty acid. So what's the other part? Yep, hydrophilic head group. So by definition, a fatty acid is amphipathic. It has both the nonpolar and polar. So we call it amphi, I believe, amphipathic, right? Okay, so this is just one type of a lipid. Fatty acid is just one type. There are many different types, but in general, they are nonpolar and hydrophobic. But it's a very diverse group, right? So if you were in your kitchen, right, and you have a fat out at room temperature, what kind of fat might you have out at room temperature? Butter. So at room temperature, what is it? RT is room temperature, it's a solid. Oil, if you have virgin olive oil sitting on a container by your stove, right? Is that a solid? No. So this one at room temperature is a liquid. Right? They're both lipids by definition, but they have very different properties. And this is just 2 examples. It's not all of them. But their composition is really important for their physical characteristics. And we're going to talk about why fats and oils are different at room temperature. But you can really give some basic classifications of lipids. Have y'all heard of lipids that are sponifiable? So sponifiable is just the ability to use to make soap. So it's SAPONIFIABLE or non, right? Sponifiable. So if you go in have you ever noticed in the lab that there are these homemade looking squares of soap by the sinks? So those were actually made in what lab? They were made in one of the labs. I don't remember now. I used to know, but I've forgotten. But it was actually made here on campus. There's a soap that is made from fats. And so if you think about it, right, how does soap work? Do you know how soap works? It emulsifies things. What does that mean? Right, so things that are amphipathic, right? They have polar head groups and they have non-polar tails. So if I get a collection of these, for example, fatty acids down there, what shape are they going to make when you have a bunch of them together? You have a polar head group and then a tail. Polar head group and a tail. Polar head group and a tail. Polar head group. What do we call this? A micelle. Like how many times have you seen in Walmart the micellar water? You know what's in there? Because the fat's in there. Put a little bit of oil in some water, you got micellar water. So why does it work? Well, because typically most dirt is non-polar, right? So you have this little bit of dirt that gets stuck in the middle of this micelle, right? And so this micelle then carries it down the drain, right? So it takes it off of your face, off of your clothes, off of your hands, and just sends it down the drain. It doesn't actually dissolve it. It doesn't get rid of it. It just transports it, right? And that's what a lot of fats do inside of your body. They're transport molecules. And that's so, God, I take so many side trips. This is why lectures take me so long, but it's so interesting. This is one of the ways that they're making some of the vaccines now, right? Because we know in the bloodstream, different proteins and cholesterol and all are transported in lipid droplets. That's what they're putting the mRNA from the vaccines into a lipid that your body knows what to do with, and your body, the cells of your body will take it in. And that's how the vaccine's effective. It's really cool. It's really cool. All right, so you can classify fats, so much of a side trip, as sponifiable or non-sponifiable. So non cannot be made into soap. So our examples of them are cholesterol, prostaglandins, all those kinds of substances. So these are examples of lipids that do not contain fatty acids. So they do not contain, I was writing not, fatty acids. Right, this is an example of a cholesterol molecule, right? It doesn't have the same hydrocarbon tail and hydrophilic head group. You do have non-polar and polar portions of the molecule, right? We have an OH here, right? That's going to be polar. We have a long, not a long, but a relatively long fat hydrocarbon tail. You have some different things in here, some ring groups that are relatively non-polar. So you do have similar characteristics, but you don't actually have fatty acids in all lipids. That's kind of a common misconception, that all lipids contain fatty acids, and they don't. But the ones that can turn into soap and wash away your dirt particles and stuff like that do contain fatty acids. And these fatty acids are long chain, that we were just talking about it, long chain hydrocarbon tail. And then they also have, what do we call, what's inside of this hydrophilic head group, right? There's a particular functional group, carboxylic acid. That's why we call them fatty acids, right? I guess I put that in parentheses, huh? I should have said this group is polar. things I need to go back and edit. Okay, so fatty acids tend to have greater than 12 carbons. So that gives them that really, really long hydrocarbon tail there. And the most common are between 16 and 18 carbons. So they're really pretty long. That's greater than, that's a greater than sign.
712 carbons.
Greater than 12 carbons. I saw the squinting. I was like, oh, okay. So you got to think about why fatty acids are important for cellular metabolism, right? So normally, right, cells are going to take in glucose. That's their primary fuel source, right? And what are they going to do? They're going to metabolize it, and we'll get to this in Biochem 1. We'll talk about metabolism of glucose. And so you go through glycolysis, then what do you go through? Krebs cycle, and then you go through electron transport chain. And what do you get at the very end? ATP, right? So you have to do this in the presence of oxygen, and you're going to get ATP out of it. So that first real conversion is to take glucose and convert it to pyruvic acid. And so then pyruvic acid is going to enter the citrate cycle, citrate or Krebs, however you want to talk about that. I like saying the citrate cycle because it tells me some information about it instead of saying Krebs, because that's just a person's name and I don't believe in that. I hate that. Things that Dr. B hates: hydrogen bonding and naming things after yourself in science. Crap. So anyway, so it enters the citrate cycle. Then we're going to go to the electron transport chain, and then we're going to make ATP. And ATP is our energy for the cell, right? But what happens if your energy needs are already met? Like you have lots of energy inside of the cell. What are you going to do with that glucose, with that pyruvic acid that you got from the glucose? We're going to store it, right? Because our bodies are trying to prepare for winter. Winter is coming, right? Our bodies don't know that we have shelter and we have heating and that kind of stuff. So it's all about survival. So we prepare for that by using these 2-carbon pyruvic acid. This is 2-carbon molecules, right? In pyruvic acid. We're going to use those 2-carbon molecules to make fats, right? Winter is coming. We're trying to prepare. So there are a couple of different fats that you can make, right? And we can classify them. So saturated fatty acids. Why do we call them saturated? They're full of hydrogen. So if they're full of hydrogen, they must have all what? Carbon-carbon single bonds. As the carbons are saturated with hydrogens. So then unsaturated were the two possibilities. You could have carbon-carbon double or really carbon-carbon triple bonds, right? So you've heard about that on your food labels, looking at saturated versus unsaturated fats. Now that you could really have a debate on how much of each you need and the sources, and that's a really, really interesting conversation. But there are two other classifications for fats. You've heard of cis fats and trans fats. So mostly you only hear about trans fats, right? Okay, and the reason why is because our bodies have never naturally seen trans fats, ever. Our enzymes are not designed to use trans fats. That's why they don't want us to eat them. And it's true, our bodies don't know how to metabolize them. So if they can't metabolize them, they're just going to pile up, right? And so that's why you end up with cardiovascular issues that go along with eating a diet that's high in trans fats, right? There are some places where they're trying to ban completely trans fats from food, right? But so And I'm going to show you how trans fats are formed and all this kind of stuff. But why, if it's so not good for our bodies, why do food manufacturers have trans fats in their foods? Shelf life. Shelf life. It lasts for a really long time, which is really important for food manufacturers, right? So you have to, like, you know, you should know all sides to the story, right? So, yeah. Okay. So, so in cis fatty acids, anytime you have either a cis or a trans, you're going to have either a double or a triple bond, right? But in the case of cis fatty acids, sorry, the hydrogens attached to the carbons in the double bonds are, what does cis mean? Same side, are both above or below the double bond. Above / below the carbon-carbon double bond. So this is like the plane of the bond. And we're going to look at some examples in a second. Trans fatty acids, right? We know trans means one up, one down, right? So the hydrogens attached to the carbons in the double bond are arranged so one hydrogen is above the plane of the carbon-carbon double bond, and one hydrogen is below. So you've looked at cis and trans of different molecules, right? Before? Yes. Okay. So here's an example of a saturated fatty acid, right? So can you have a trans or a cis saturated fatty acid?
No.
No. In order to have cis or trans, you have to be unsaturated. So if we look at this molecule, really there are two different possibilities. You could have both hydrogens being up, and in that case, and it doesn't matter, you flip the molecule right up or down, it's the same thing. But they're both on the same side of the double bonds, on the same plane of the double bond. And trans, we have it on opposite, right? So naturally, you don't form these. These are not found in the body. These are not found in animals that we eat. They're not found in plant fats. They're just not found in nature. So our bodies naturally make cis fatty acids. And my abbreviation for fatty acids is FA. So we have enzymes that can break down those cis fatty acids, right? The most common cis fatty acid in our natural foods is oleic acid. So in all of our meats, our beef, chicken, pork, our cheeses, our nuts, all the things that we're really commonly eat, a lot of it is oleic acid, right? So we know how to process that. Our bodies are accustomed to that. The trans fats, on the other hand, are created in the processing of the foods, right? So in food processing, the fats undergo hydrogenation and dehydrogenation rounds. Hydrogenation, dehydrogenation. I don't know why it gets me that little thing on top. That's weird. So when you do this, right, dehydrogenation, hydrogenation, what are you doing? You're taking hydrogens off and you're putting hydrogens back on, right? So you've heard of food that is partially hydrogenated, right? It's that partial hydrogenation that results in trans fats. So if it says partially hydrogenated, whatever kind of fat on the ingredient label, it's got trans fats. It does. So you start with something that has a double bond to begin with. It's in the cis orientation because that's how all fats are found naturally. And then you go through this hydrogenation and dehydrogenation cycles. And so one of these hydrogens will come off, right? And pop back on. And so what ends up happening is you have some fats that become completely saturated, and you have some fats that are still unsaturated, but because the hydrogen came off and then came back on in the opposite orientation, you now have trans fats. So you start with something that is a cis unsaturated fatty acid, and then you go through a hydrogenation process, but it's not complete. So some of your fats are completely saturated and you have no more carbon-carbon double bonds, and some of them are still partially unsaturated, but the hydrogens have popped off and popped back on in that trans configuration. And we don't have the enzymes. We just don't have the enzymes to break down the trans fats. So that's why they're bad. But does that mean I don't eat them? Yes, I eat them. I'm sorry. I should not, but I do. All right. So fatty acids are typically not found as free fatty acids. Typically in our bodies, they're esterified to other molecules. Esterified. Esterified. Right? So what happens in esterification? So if we have a fatty acid, in that fatty acid, we have our carboxylic acid, right? So here's our 3 fatty acids. And do you notice how I just put an R here? Because really what I care about in this esterification is our carboxylic acid. So you recognize that as carboxylic acid, right? COOH, it's just another way to write it. And so what we do is we have an alcohol on a glycerol molecule, right? So we have our OH. You see how each glycerol molecule has 3 OHs on it, 3 alcohols. And I'm going to take 3 fatty acids and I'm going to undergo an esterification reaction. And what's going to happen is that I'm going to lose this hydrogen, and I'm going to lose a hydrogen and oxygen, and I'm going to end up with, what do we call this? This is a triacylglycerol, right? So 3 fatty acids hooked to 1 glycerol molecule. That's why they call it tri for 3. 3 tells us 3 fatty acids, and glyceride means that it was derived from glycerol, right? Okay, so this is, so what do we now have? If you have a carbonyl carbon bound to another oxygen, you have a what? An ester. That's why we call it esterification, because we now made an ester. Yeah, kind of cool, right? I think everything's kind of cool if it's science, but it is. It is. All right, so you can have triacylglycerides. That's entirely possible. But you can also, instead of attaching fatty acids here, you can attach other things to these oxygens. So you can have a triacylglyceride. So this is my tri, Y-C-E-R. I have to say it as I spell it, I'm sorry. So what do you think the Mg and the Dg stand for? Mono and di, that's exactly right. Monoacyl, A-C-Y-L. Monoacylglycerol. Diacylglyceride. Okay, what?
Is it glycerol or glyceride?
Glyceride, triacylglyceride is what you make. You derived it from glycerol.
Okay, because you put monoacylglycerol.
Glyceride, I-D-E, sorry. Glyceride, I-D-E. This is terrible. Yes, ma'am.
So triglyceride and triacylglycerol is the same.
Same thing.
Do you prefer?
Doesn't matter. Doesn't matter. So different fats are used in different commercial processes. So diacylglycerides, those are really, really effective in keeping other fats in solution. So think about ice cream, right? That's my favorite example of it. So it keeps all those fats really well suspended. Triacylglycerides are really high in energy. So we want to use these in our bodies for energy storage, not in cell membranes. We really want to keep it in our adipose tissue. So if you've ever seen, gosh, that comes out really dark. Can you hit the lights for me in the back? So if you look at these are adipocytes. So have y'all looked at any adipocytes in the lab? I know a few of y'all have in high school. So this, no, it's not onions. Yeah. So these are each one of these circles, right? You would think that that's a cell, but actually there is one cell and it's on the outside of it, and almost 99.9% of the cell is filled with one lipid droplet. So there's like a vesicle, and inside that vesicle is all the fats, and everything else in the cell just gets smooshed and smashed against the plasma membrane. And so can you see that there are some really dark dots? Those are the nuclei. They're like smashed, right? So you can't even tell, is this nuclei for this cell or is this nuclei for that cell? You can't tell because they're all just smashed, right? Because they're filled with these lipid droplets. And inside of those lipid droplets, we have the triacylglycerols, right? Okay, so here we go. Now let's talk about what we're actually going to purify. The ones that we're actually going to purify are phospholipids. Where do we find phospholipids? In the plasma membranes. So notice, here's our glycerol backbone, right? So we have an esterification right here. So we have our ester here, we have an ester here. So we have two fatty acid tails. What did we attach to the third? We attach some kind of a phosphate group. So you see how between that carbon, the oxygen that was in the glycerol, then we have, this is a phosphate, a charged phosphate group. And then do you see the teeny tiny R, right? There are lots of different things that could be at that R position. So depending on what group is there tells you what kind of phospholipid you have. So, so when we make a phospholipid, we're going to have a di, that's supposed to be a D, not a very good D, diacylglycerol, right? Because we want 2A, what are we putting on the top here? A phosphate group, 2A, phosphate. Conversion of a diacylglyceride to a phospholipid, pH. Oh my gosh, I cannot spell today. Right, we're converting our diacylglycerol to a phospholipid, involves the addition of a phosphate group to the glycerol via, in this case, it's not going to be an esterification reaction, it is going to be a condensation reaction. I'm just going to go with it and keep it. It's a condensation reaction. So there are many different variations of our phospholipids. Typically, what you have here, the R1, right? R1 is going to be a really long tail. R2 is going to be a really long tail. And then you're going to have your phosphate attached to that third oxygen of the glycerol. And then this X group is going to vary. So depending on what group you have put in what position tells you what kind of phospholipid you're going to have. And each tissue has a unique combination of these different phospholipids. So heart tissue has a particular percent of each of these different phospholipids, whereas liver tissue would have a different percent of each one, right? So we're going to focus our experimental question is really, of all these phospholipids, what is the percent of each in heart tissue, right? That's our question that we want answered. Do you think that answer is actually out there? Yes, so for this next experiment, I'm not going to tell you what your percentages should be. You are going to run the experiment. You're going to find out for chicken hearts what the percent of each of these phospholipids is. Then, instead of doing calculations and additional problems, you're going to go into the literature and you're going to go find some citations where they actually talk about cardiac lipid composition, right? What's each percent? Now, are you necessarily going to find chicken? Maybe not. What might you find? Beef. You might find mouse, right? Mouse is a really common animal used in research. You might find human. It doesn't matter to me. Just find a mammal, right? Be pretty similar enough. Mammal's going to be similar enough. Yeah, yeah. You start changing the chemistry of the body system, you're going to start changing the lipid percentages, right? Okay, so how are we going to do this? We are going to do what's called thin layer chromatography. So what's chromatography? We talked all about in class with column chromatography, right? So are we going to use a column for this? No, we're going to use a very, very thin layer of silica gel on a glass plate. And the lipids will travel at a different rate along this plate due to their actual charge. So, so this is why I put mammalian heart tissue, because sometimes we do different species, different, you know, whatever we can get our hands on, basically. So anytime you do thin layer chromatography, it's really a three-step process. The first step is to actually take your sample. So you're going to have to get the hearts. You're going to have to basically homogenize them, right? In some way, shape, or form. Remember, we talked about that when we were doing protein purification. First, you have to break open the cells, and then you have to actually separate. I want to get rid of the proteins, right? Do we want proteins for this experiment? No. So we actually want to get rid of those proteins. So I'll show you how we do that, so that we can really just get the organic lipid portion of all of those cells. And so what we're going to do is we're going to put that lipid sample into a very volatile solvent, right? So the great thing about something that's volatile is that it evaporates super quickly. So we're going to put it at the very end of the TLC plate. Then the solvent will evaporate, and then I'll have a solid lipid sample at the bottom of my plate, right? And then we're going to put this plate into a pool of another solvent. And that solvent is going to travel up my plate through basic capillary action. Well, what's going to happen? It's going to pick up those lipid particles and it's going to carry them along the plate. So it's going to move, right? And so what happens is that your spot of sample is going to travel up the plate, but it's going to depend on the chemical composition of the lipids inside your sample as to how quickly they will travel up that thin layer chromatography plate, right? So it's really important that your development solvent be polar, right? Because remember, like dissolves like. So if I want my sample to actually move and not be stationary, it has to be dissolved inside of the solvent. So if my phospholipids are polar, are they polar? We have polar head groups. We have polar head groups. So they're amphipathic. So we have some polar, and depending on the head group can make it more or less polar. It will actually interact differently with the solvent, which will make it migrate differently on the plate, right? So at the end, we'll have different spots, and I'm going to show you an example of that. And the very end is to visualize what's there. So we're going to incubate our plates with iodine, and the iodine is going to have a reaction with the lipids, and you'll be able to see them as a dark yellow spot. So this is basically what you'll see at the end of the experiment. This is somebody from, I don't know what semester. I was like, oh, I need a picture of that. And so we just took a picture of their sample. So what you end up doing, this is the very beginning, right? We spot our sample on basically the bottom of the plate. We put the plate into a tank that has solvent. the solvent will, through capillary action, go up the plate, and it will pick up our samples, and they will travel at different rates, right? Up that silica gel. And then when we get to a certain point where we say, okay, that's enough, we can take it out, we let it dry, and then we can measure the distance that it traveled and determine which lipid it really is. So there are 7 different samples, 7 different bands that you should see. So I have them labeled here. See at the bottom, number 1, right? Number 1 is lysophosphatidylcholine. So these are the ones that you're going to want to go and research, right? Because I'm telling you on the front end that these are the ones you're going to be able to extract from our mammalian heart tissue. So I'm kind of giving you a jump on the gun here. The second one, very light. Do you see how it's very hard to see these first 2? They're going to be very, very light. So sphingomyelin. Can you predict if you're going to have a lot of sphingomyelin in your heart tissue? Not so much, right? Where do you think you might have a lot of sphingomyelin? What's myelin? It covers your nerves, right? So if I were looking at brain tissue, then I would expect to see more sphingomyelin, right? So different tissues are going to have different concentrations of these different phospholipids. PC is going to be phosphatidylcholine. So do we have a large band of phosphatidylcholine? Yes, we do. Band number 4 is a combination of 2. They run at the same place because of their polarity. You have phosphatidylserine and phosphatidylinositol. And so that's band number 4. It's kind of that lighter band. Then we go up to number 5. It's phosphatidylethynolamine. So that's going to be a relatively dark band. Then you have phosphatidic acid, very, very light band right over here. Then you have phosphatidic acid, number 7. Now, at the solvent front, this is how far we let the fluid travel up the silica plate, right? What you're going to end up having is nonpolar lipids, right? Those are going to be at your solvent front. So I think that's why we labeled it SF, solvent front, right? Okay, so, and I said that nonpolar lipids, which are in the solvent front, and the PC and PE should be the largest bands. So those give you really good indicators to help you number it, to help you figure out which ones want what. We do not want the nonpolar lipids. We only want the polar lipids. So do not collect the solvent front. Okay, so put yourself a little note, do not collect the solvent front. So remember how we said that to extract those lipids, we want to get them to come out. So we want a nonpolar solvent. So the nonpolar solvent that we're going to use is a combination of methanol and chloroform, right? So one group is going to go in and blend all of our heart tissue together inside of methanol. Then they're going to add 100 milliliters of chloroform to a final ratio of 2 to 1. Then when they're done, right, while they're doing that, I guess, actually, while they're doing that, then the other groups are going to skip ahead and they're going to start preparing their tanks, right? So y'all are, whoever decides to blend, the other groups are going to kind of help out that group, right? So if somebody gets to make the smoothie. Yeah, right. It's what are they, Smoothie King Friday, right? Eat your $5 smoothie. Nobody's gonna want to go get smoothies again. So you're gonna have to prepare the solvent. So see this blue solvent here? That is going to be required to run, because you actually have to make sure that you have the same humidity in the tank for the whole time of the run. So you have to make the solvent early, put it in the tank, put a lid on the tank, and let it equilibrate. Otherwise, what you end up having, if this is your plate, if you don't let it equilibrate and you put it in right away, you're going to have varying amounts of travel for your solvent, and you'll end up, instead of with a straight line, you'll end up with like a smile. And it's going to be really hard to see which band is which because they'll be all kind of mixed up. So you don't want to do that. So that's why equilibration is super, super important. Okay, and I think I tell you how to do that down below, right? So then at the end of all the blending and all that kind of stuff, each group will get 30 mils from the blender. So we're just going to actually process it in a blender. And they're going to put it into a purple top centrifuge tube. What we want to do is we want to separate lipids from our homogenate. So we're going to mix it with a certain buffer. Our buffer, what does PBS stand for? What buffer? Anybody? Phosphate buffered solution, right? And so it's actually going to create gas when you do this. And you're going to have this in a tube with a screw top. So if you shake it and you're actually making gas, what's going to happen? That gas is going to build up. So you have to unscrew the top, let out the air, and then screw it back. So we call that burping. You burp your tube. You know, I've done before, right? So do it a bunch of times. I don't know, 6 to 10 times, something like that, right? And so what you'll end up happening is that the homogenate will separate into phases. There'll be 3 phases. There'll be a top aqueous phase, and that'll be our proteins, right? Our soluble proteins. Do we want that? No. There'll be a middle interphase. It's going to be relatively small, and it'll be white. And you'll see that you'll have denatured proteins in it. And then at the very bottom, you'll have the organic phase, which is going to contain our lipids. But you might not see these 3 phases right away. So if we spin it for 5 minutes, you'll see a very nice separation between these 3 layers. Some people get it right away and some people don't. But if you spin it, you'll get plenty. So which of these 3 do we want? We want the bottom organic phase. So this is what we want. So we're going to use a pipette. And we're going to take that pipette and pierce through the top phase, through the second phase, and have that pipette enter the bottom phase. And then you suck up 5 to 10 milliliters of that bottom phase. Doesn't really matter how much. It really doesn't. 5 mils is plenty. But you'll have about 5 to 10 mils of it. Okay. And you place it into a new clean tube. And this is going to be your sample. This is your lipid sample. So while group 1 was purifying, was homogenizing the sample and all that, all the other groups are going to place a sheet of chromatography paper, which I'll show you when we walk over in the lab, and pour your solvent in and allow that humidity to equilibrate so you don't get that smiling. So this is how you prepare the TLC solvent. This is super important. This is super important. Order is important. If you put it in out of order, it's going to precipitate. Okay. So what you do is you start with water and you slowly add methanol to the water. And you're constantly swirling as you're very slowly adding methanol to the water. Then once all the methanol's in, then add chloroform while constantly swirling to mix, right? So here are your ratios. You should have 25 milliliters methanol, 4 milliliters water, and 65 milliliters of chloroform. At the end, when you add all these 3 things together, it should be clear. If it's not clear, it will not run properly. You need to start over. Okay, so if you want me to like come and look at it, but you can tell. Trust me, you can tell. But if it's not clear, take the time, stop, remake it. Okay. All right, so you pour your made solvent into the TLC tank, you cover it, and you let it equilibrate. Now, one thing that's really, really important, I'm going to scroll up here. Do you see how the solvent level is lower than where we spotted the plate? If your solvent level goes above where you spot the plate, what's going to happen? It'll just float off. So we don't want that to happen. So just make sure that when you're putting your plate in, that you went up high enough. And if you follow my instructions, you will not have that problem. Okay, so after your tank is prepared, then you need to prepare your plate. So on the plate, you're going to use a pencil and you can actually write on the plate. And I'm going to show you when we go in there. You write top and you put your initials so we know which group has what plate, right? And you're going to draw a line that separates into left and right plates. So we'll have 4 groups and we'll have 2 plates because, right, 2 groups can share a plate. So at the bottom of the plate, you're going to make a light dashed line that's 2 centimeters from the bottom, and you're going to go straight across the bottom. So it'll look like this. It'll just look like a straight line across the bottom of the plate. And on that line is where you're going to spot your samples. So you put a couple of drops, you let it dry. You put a couple of drops, you let it dry. You put a couple of drops, you let it dry. And so you keep layering onto the same spot so that it gets dark enough. And then it's really important to put drops and then skip enough space so that your two samples don't overlap. Otherwise, all your calculations are not going to work. So you use your Pasteur pipette. Anybody use a Pasteur pipette in the past? It's a very, very thin tube. I'll show you in there. No problem. And I'll show you how to do that. And then you allow your plate to completely dry. Then you put your plate into your tank. Don't put it in at an angle because then you're going to lose the sample at that corner. Put it in as straight down as you can. And we'll run it for probably about 30 minutes. So we'll have about a 30-minute run time. You have to stay in lab, but we'll have time. So you remove your from the tank, you let it dry, and we're going to incubate it in an iodine chamber for about 5 minutes. And that's where it will turn this beautiful yellow, muddy looking color. Right? Okay. So now that you can see the bands, I want you with a pencil to label the fractions, right? Label it just like we did here. 1 is LPC, 2 is SPH, right? Label them. And if you need to label them and go back and put it in the iodine chamber again, you can. It's not a big deal. But just make sure you label it really, really well. All right, and then you're going to scrape off one band at a time. And so you scrape that one band off onto a sheet of whey paper, and then you use that whey paper to fold it up nicely and pour it into a glass test tube. And so you should have how many test tubes at the end of this? You should have 7 test tubes at the end of this. There'll be an 8th band, but are you going to collect it? Yeah, okay, good. Awesome. All right, so now what we want to do is we want to be able to quantify the amount of phospholipid there. But we don't have a technique to measure lipids. But we do have a technique to measure phosphate, amount of phosphate. So what we're going to do in the very last step is right here in number 10, is we're going to use perchloric acid to basically chop off the phosphate group head to remove phosphate head from phospholipid. But this is going to have to cook in the oven for 2 hours. So guess who's staying in lab 2 hours after y'all leave? Do you have to stay for the 2 hours and watch it cook in the heater? No, you don't. I'm going to let you go home, and I'm going to stay, and I'm going to watch them, and I will pull them out when they're done. You're welcome. I thought that might make your day. So if you're going to put it, we have to put it in a heater for the perchloric acid to work the best. So you can't put anything plastic or it will melt in the heater. So you can't cover it with anything plastic. So we cover it with a glass marble to prevent evaporation, but at the same time, nothing can melt, right? And I'll show you where those are and stuff. And then in the next lab, we'll quantify the amount of phosphate into each sample. So do you have a lab report to turn in after that first day? No. You're going to have to come in and do day 2, and then you'll have a lab. But what could you be doing right now?
The background.
You can start your background. You could do your literature search to find phospholipid percentages and all that kind of stuff. Yeah. Okay, so we're going to walk next door for like 5 minutes just so I can show you the supplies, and then. Yes, I do.